Scientists try to help large and smallholder farms go beyond practices for sustainable agriculture to regenerative ones. This presentation describes portable elemental analysis and its applications to help meet those goals. Regenerative agriculture's primary goal is to enhance a given ecosystem. This includes practices to naturally enhance soil and crop quality, to boost the resilience of crops, to reduce unnecessary inputs such as excess fertilizers, to regain the nutrient density of food, and to restore the fertility of exhausted soils. These practices can help to mitigate the impact of changing climates on soil and crop health, including the ability to retain and sequester carbon dioxide and to increase the capacity of soils to hold more water. Let's first review the physical components of a plant system. It starts with quality seeds, which are high in germination and free from insect or physical damage. They are placed in planting media such as soil, peat moss, coconut coir, rock wool, perlite, vermiculite, or even an aqueous solution. Inputs are an important aspect of a plant system. The needs vary depending on the plant, the condition of the media for that plant, and the overall environment the plant is grown in. Of course, plants need water to ensure their physical ability to obtain nutrients. The root system helps deliver water and minerals to the shoot system, which is what we typically see and access for food. Key environmental factors which affect a plant system include temperature, water and humidity, light, soil pH, and the mineral nutrients. In the pursuit of healthy plants and soils, many elements are of interest, and since growth is a varied and dynamic process, elements need to be monitored at various stages. Macronutrients are typically found in the percentage levels. Micronutrients are most often found in the parts per million levels. Other elements of interest are also usually in the parts per million levels. There are metals that are detrimental to crops. Hopefully, heavy metals and other dangerous metals are not present, but in some locations they can be found at high parts per million or even percentage levels. Fortunately, key environmental factors can be measured using field portable devices. Most people are familiar with measuring temperature with a thermometer, humidity with a hygrometer, pH with a pH meter, and light with a light meter. But not everyone is as familiar with elemental spectrometers, which are used to measure mineral nutrients and heavy metals. The portable elemental analyzer technology highlighted in this presentation is X-ray fluorescence spectrometry. That may sound complicated the first time you hear about it, but it's a straightforward, fast, and accurate technology used to find out which elements on the periodic table are in a given material. Let's look at a quick demonstration of a soil measurement. The soil application is selected and the analyzer window is placed in direct contact with the soil before pressing the trigger. You can see that this soil has percentage levels of iron, calcium, and potassium, and that there is about 35 parts per million of arsenic. Natural levels of arsenic range from about 1 to 40 parts per million. However, it would be a good idea to do further analysis before using this soil for edible plants. Before we discuss specific XRF applications, let's look at how XRF works. It can be compared to how we sense color. Most of us can see visible spectral colors from red to violet. If we look at an electromagnetic radiation diagram, we can see that visible light photon energy ranges from about 1.65 to 3.10 eV. That means when the sun or other light source shines on an object, our optical system detects photon energy signatures of color from about 1.65 to 3.10 eV. For example, we determine that these corn plants are green because processors in our brain know that the photon energy signatures we detected from 2.25 to 2.34 EV are green. Let's imagine the XRF analyzer window as an eye which looks at objects or samples to detect X-ray photon energy. It can see X-ray energy signatures from about 1,000 to 40,000 eV. However, instead of seeing these photon energy signatures as colors, it detects them as elements on the periodic table. Like colors, signature energies of the elements on the periodic table are well defined. For example, bromine has a primary signature energy at 11.9 keV. An XRF can detect photon energy signatures of magnesium at 1.2 keV, phosphorus at 2 keV, potassium at 3.3 keV, iron, nickel, zinc, arsenic, mercury, and lead at their signature energies. When the X-ray tube shines on a sample, the XRF's optical system and processor distinguish elements in the form of a spectrum. Each element it detects appears as a peak along the energy axis. The intensity, or number of photon energy signals, of each element's peak helps determine its concentration. The simplified answers appear on the results screen. Let's recap by using the simple illustration of a corn crop experiment to better understand. Two different types of corn seed were planted and grew. When the sun shines, most humans detect two different shades of green, from about 2.25 to 2.34 eV. If we let the XRF shine an X-ray tube on samples taken from each cornfield, it detects more photon energy signals of aluminum, silicon, and calcium in the dark green field, and it detects more phosphorus and potassium in the light green field. Both views detect significant variances in the fields from two different seed types, but they see them from different perspectives. It's important to know what elements are naturally present and which ones are introduced during the grow to make informed decisions for regenerative precision agriculture. Multiple scientists use portable XRF to do just that. The following slides highlight some of the applications from their work. Plant growth starts with competent seeds to develop into healthy plants which bear more high-quality seeds. The study of bio-fortified seed development by scientists at a university and a global outreach organization in Australia provides a good example. Their primary goal is to reduce malnutrition in developing countries by breeding high-nutrient crops, especially with increased zinc and iron. They want to help minimize the need for fertilizers by instead developing seeds which increase nutritional quality in staple foods, such as wheat and rice. Portable XRF is helping them track seeds which yield nutrient-dense grains faster and more accurately. They also use portable XRF to confirm that seeds labeled as bio-fortified at supply stores are in fact bio-fortified with micronutrients and not just priced as such. These scientists utilize both laboratory and portable XRF to develop successful bio-fortified seed technology. Healthy soil is important to hold roots and store minerals, metals, and water. A consultative group in Kenya with multiple global partners operates a network of soil plant spectral diagnostic laboratories around the world. One of the first things they participated in was the development of soil nutrient maps throughout Africa to improve the regenerative agriculture potential of smallholder farms. They develop and disseminate low-cost portable elemental analysis methods for plants, soils, and fertilizers to enable evidence-based agronomy. They also support farmers with limited resources in challenging climates to ensure they are supplementing native growing conditions with the correct fertilizers and other additives. Their portable methods enable quick testing of the samples wherever and whenever they are needed. For those fortunate enough to have laboratory resources, portable XRF methods developed supplement the low-capacity, expensive, slow, and destructive ICP lab testing. This consultative group has a fleet of handheld XRF analyzers in constant use for clients around the world. You can directly measure soil with portable handheld XRF. However, some scientists prefer to collect soil samples in baggies. Either way, a quick screening provides an idea of the general composition of the soil. This is especially important for monitoring high levels of heavy metals. Sampled soil can also be placed directly into a sample cup for measurement. However, lab-like sample prep should be used to obtain the most accurate and precise measurement of soil samples. They should be dry, homogeneous, crushed into small particles, sieved, and packed tightly into a sample cup with a flat surface, following methods like US EPA 6200. One thing field scientists should keep on hand when measuring unknown soils is 1 or 2 reference samples with known concentrations. This helps ensure the overall measurement technique is in good working order. Most portable XRF manufacturers provide reference standards, but they can also be purchased from other resources or developed internally. The important thing is to have some known samples on hand in case questions come up. Another thing field scientists should have on hand is a blank silica sample to ensure each measurement is representative only of the sample being tested. Many field scientists use baggies over the nose of a handheld to minimize carryover from one sample to the next. That also helps increase the lifetime of the window on the nose of the analyzer. Another application we'd like to highlight is treatments. A large dairy farm joined forces with a fertilizer and testing company to figure out a way to turn the problem of farm manure pollution into a solution for food crop farmers. The world has a lot of dairy farms. In the United States alone, about 400,000 million pounds of cow manure are excreted every year from this industry. This turns into excess nitrogen and phosphorus, ending up in water sources, which in turn create significant problems for the environment. In addition to mitigating the damage from excess deposits of nitrogen and phosphorus, they wanted to figure out a way to recycle the cow manure into useful products. They developed a process to recover the nutrients from the cow manure and to formulate it into custom micronutrient-rich granules using minerals such as zinc and copper for special fertilizer blends, which enhance nutrient efficiency and crop uptake. This successful endeavor provides farmers with carbon-based fertilizer formulations to sustain healthy plants and soil while helping the environment. They use XRF and infrared technology to monitor incoming materials as well as for the development of various fertilizer blends. Fertilizers are quite varied in base material and content. Bruker is working with a US-based nonprofit organization whose goal is to provide solutions to create a food-secure, environmentally sustainable world. Together, we are developing a robust factory-ready calibration for portable XRF analyzers covering macro and micronutrients as well as heavy metals in fertilizer samples. Although there is no intention to put heavy metals in fertilizers, they can be present if starting materials contain them. A custom calibration is now available, which has very good correlation to ICP measurements for elements such as phosphorus, calcium, nickel, arsenic, and more. Our next highlighted application focuses on root systems. Characterization of the productivity of apple rootstocks is critical to the USA's 7,500 apple producers and $4 billion industry. The ability to predict which minerals from a root system accumulate in the shoot system is an important aspect of work being done by a collaboration between the U.S. Department of Agriculture and a local university. Their research focuses on ways to develop new apple rootstocks that are more productive than current commercially available varieties and are resistant to devastating diseases like fire blight and replant disease. They use portable XRF to understand which nutrients accumulate in the leaves, which in turn helps them identify key genes to improve the root-to-shoot process. One method they're working on concerns predicting disorders which are believed to be induced by deficiencies in certain minerals. They use handheld XRF to study these processes. One of the methods they developed is to monitor potassium to calcium ratios. They have found that these ratios are able to predict bitter pit, which adversely affects the value of produce such as apples. Research from another horticulture group involves measuring ratios of elements to the XRF's inherent rhodium signal as an internal standard. Elements such as calcium, chlorine, and sulfur are used to help preserve produce as it travels from the farm to the grocery store. Monitoring these ratios helps optimize surface treatment processes to make sure the entire fruit is coated, thus preserving the food during transport. Now we'll look at plants. Plants rich in nutrients and free from heavy metals provide healthy food and other ingestibles. Multiple universities around the world use portable XRF to teach students about plant uptake, to research plant ecosystems, and to reach out to the community to aid in more productive local and urban farms. Many professors find portable XRF to be an ideal teaching tool, not just for university students, but also for STEM outreach programs to local schools. Since everyone can relate to plants and portable XRF is so fast, they can get students to engage quickly with the technology. This then gets students to ask more high-level questions, such as, what is the environment the plant is in that results in its condition? The portable XRF then acts as the tool to help them find the answers. The use of plants for bioremediation is also being researched. Studies are conducted to help determine which plants are optimum to take up which dangerous metals. Elemental analysis of the plant tissue helps determine its effectiveness. An important research project for regenerative agriculture includes the ability to correlate elemental accumulation of leaves with differences in root length. One university is focusing on maize roots and the availability of deeper roots to sequester more carbon in the soil and to decrease the need for nitrogen and water without compromising the leaf's ability to accumulate nutrients. Another university is using portable XRF to reconstruct the diets and habitats of extinct species. They do this by comparing patterns and data from known modern habitats to similar data preserved in the fossil record. They are quantifying variations in strontium to calcium ratios in fruits and leaves, as well as in teeth and bones to base the reconstruction on. Multiple universities use their portable XRF to help local and urban farmers improve their grow conditions. Some focus on determining how heavy metals get into plant systems, such as cadmium in spinach grown near old battery plants. Others focus on enriching nutrients in plants to improve plant-based food sources. Universities use a mix of handheld and countertop portable XRF analyzers to support this work. Most plant tissue is cellulose-based, which proves to be a clean matrix for building a universal XRF calibration. Many people want to do live plant analysis, which is possible, but the relativeness of the data must be understood. For accurate and precise XRF quantitative analysis, especially when correlating to ICP results, samples should be dry, flat, homogeneous, and infinitely thick. Plant tissue does not exhibit any of those characteristics. In addition to being thin and having uneven surfaces with stems, veins, and pulp, plants contain substantial amounts of water. Using proper sample preparation procedures, Bruker developed a robust factory-ready calibration for its portable XRF analyzers covering nutrients and heavy metals in plants. Studies show that plants can have up to 95% water in the leaves, which will adversely affect quantitative analysis if not taken into account. Therefore, it is critical to properly prepare plant samples for quantitative analysis, especially if you are using a calibration whose reference materials were prepared properly. It is not as complex as preparing a sample for ICP or other wet chem analyses. In fact, all you need to do is dry the sample in air or protect it on a hot plate, crush and homogenize the sample in a simple food processor, and then pack the sample tightly into a sample cup. We hope you enjoyed learning how portable XRF works and how some scientists are using it to analyze for nutrients in seeds, monitor nutrients and heavy metals in soil, measure major and trace minerals in fertilizers, as well as screen them for heavy metal contaminants, determine mineral accumulation from the root system, and analyze plant tissue for nutrients and heavy metals. Bruker has several portable XRF configurations available for elemental analysis in the lab, greenhouse, back of the truck, or in the field. All can be used for qualitative spectral analysis, which is greatly enhanced with RTAC's PC software. Multiple factory-ready calibrations are available to be loaded onto any of these analyzers, including one specific for nutrients and heavy metals in plants, fertilizers, or soils. Our application scientists are also available to create and load custom calibrations on the analyzers with customer-supplied reference materials. Additionally, customers can create their own calibrations using EasyCal PC software and load them onto the analyzers. If you'd like more information about how XRF can work for your applications, a budgetary quote, or to schedule a demonstration, please contact our sales department at sales.hmp@bruker.com or at 1-509-736-2999. Thank you for your time and attention. Please take care and stay safe.